Second half today, what I want to do is concentrate on the particular functions of digestion, pretty much following through the digestive tract. So I'll talk a little bit about the mouth, pharynx, and esophagus, and then get into the stomach and the small intestine and a little bit about the large intestine as well. And then I'm going to go up to this section on the liver and pancreas and gallbladder. And I'm going to be using the book primarily. The stuff that I was using out of the presentation earlier was sort of specific about that enzyme stuff, but here I want to just refer to what the book's going on about. So in this first section, mouth, pharynx, and esophagus, like I said earlier, it's mostly about chewing and swallowing. Those are the two main functions that happen here. And so here we have a figure basically pointing out most of the structures of the oral cavity. There's a second, and followed by the tongue, salivary glands. This is where I thought I was going next. And then a section on the teeth. In the center, they have the so-called baby teeth, and then the adult teeth that grow in after, listed there. And a little description of the anatomy of a tooth. As far as specifics for dental hygiene, this is kind of the main place where it comes up. I'm not going to cover any of this stuff. If you're headed for dental hygiene, I just want to direct you to this part of the chapter that's addressing teeth. They are also addressed a little bit in A&P1 in the skeletal system stuff. The book has in several places tables like this that list the various functions of structures in that part of the book. Okay. So these are for the oral cavity. I want to remind you here in the pharynx, just to point out, and this came up in the respiratory system part, the pharynx has three different regions: the nasopharynx, which is adjacent to the nasal cavity, the oropharynx next to the oral cavity, and the laryngopharynx next to the larynx. The oropharynx and the laryngopharynx are part of the digestive tract. The nasopharynx is only part of the respiratory tract. Now the oropharynx and laryngopharynx are part of the respiratory tract also, but that's where the digestive tract crosses over. And then the esophagus is pretty much just a tube that connects down to the stomach to deliver the food that we've ingested at the oral cavity with the stomach. It's really just there for delivery of food from one location to the next. The movement of the food down there is swallowing, which is more formally called deglutition, and it has a few different phases. We could say that it has two major phases, the voluntary phase and the involuntary phase. And then the involuntary portion is split between the pharynx and the esophagus. The voluntary phase is basically moving food around in your mouth with your tongue, and when you are ready to swallow something, you move the food towards the back of your mouth. In the wall of the soft palate, the soft part of the roof of your mouth, there are sensors which are going to respond to the presence of food when it gets back there. Solid foods are mostly going to activate pressure sensors, and then liquid foods and solid foods can also activate temperature sensors there, all of which will initiate the involuntary phase or the reflexive phase in the pharynx, which begins with, let me jump back up here. Oh, I know. The picture I want to get to is, no, the picture I'm thinking of is in the respiratory system. actually, let me go to that because I do want to show you this. So this is actually from the respiratory chapter in the book. But when we push food around in our mouth using the tongue, that's the voluntary phase. And essentially, we're pushing food around to help break it down and chew it. And then once it's fully chewed, we push it towards the back. And then in the soft palate, there are sensors that respond to the presence of food. And what happens is the soft palate and the uvula, which is the sort of thing hanging off in the back of your throat, move back to block the nasopharynx, which actually this particular picture is showing that, well, I guess not. I guess this is the back of the pharynx here. It kind of looked like I thought this was the back of the pharynx. It looked like it was pushing back against that. But so the opening to the nasopharynx here will be closed off by the soft palate and the uvula so that food doesn't go up. And the esophagus, the epiglottis will fold down and block the lower respiratory tract. And then pharyngeal muscles will push the food down into the esophagus. And then the food will move on down the esophagus to the stomach. And in this, sorry, I just was in the right position. In the esophageal phase, what happens, the food gets pushed down through a process called peristalsis. So before we started, I went ahead and opened up this picture, which is from the processes, and I can't remember what the title of that section was, but the section right before the mouth, the one in the mouth and the pharynx. This is a demonstration of what peristalsis is. It's one of the forms of propulsion through the system, and actually it's found most everywhere, but the esophagus is a very obvious sort of general general example of what peristalsis is. The circular fibers of the muscularis will pinch off behind a bolus of food or a chunk of food. And the longitudinal fibers will shorten that part of the tube a little bit, which will help to push things down. And the circular fibers just keep pinching off behind it. I like to use my tie to kind of illustrate that. So my fingers represent the circular fibers, and then the longitudinal fibers, like I'm pulling the tie back, and it just keeps pushing the food down through the esophagus. That's peristalsis. When it gets to the end of the esophagus, it moves into the stomach. There is a valve right there at the joint, the connection between the esophagus and the stomach, called the gastroesophageal or lower esophageal sphincter, which is meant to keep things from the stomach from washing back into the esophagus. When it fails, when stomach acid does go back in, that's what causes heartburn. But generally speaking, that should close things off. So that's essentially what swallowing is about. Okay, it's in the oral cavity, we have the voluntary phase where we push food back. And then once it gets to the back of our oral cavity, that initiates the pharyngeal reflexes. And then the esophagus continues on and carries things down the rest of the way. So the pharyngeal and esophageal parts are both reflexive or involuntary movement of the food eventually down to the stomach. So to talk about the stomach, first I want to address a couple of issues for its anatomy. There are a few different regions of the stomach. Where the esophagus attaches to the stomach, that area is called the cardia, referring to the fact that it's adjacent to the heart. Not directly adjacent to the heart, the diaphragm's in the way, but it's near the heart, so it gets that name. Where the stomach kind of rises up, bulges up above where the esophagus attaches, that region's called the fundus. And fundus is just a general name. We see it in a few other places. where the uterus rises up above where the two fallopian tubes attach. That's the fundus of the uterus. Where the cornea bulges out from the sphere of the eyeball, that's the fundus of the eye. So it's just talking about any kind of bulging structure like that. So it bulges up above the esophagus. It's called the fundus. And then most of the rest of the stomach is referred to as the body of the stomach. And then it ends in the pyloric region, which has two parts, the pyloric antrum and the pyloric canal. The word antrum basically is a Latin word for outside room or waiting room or something like that. And so it's just the part of the pylorus that leads to the pyloric canal. Now on the inside of the stomach, what we see here in the lighter color, there are a bunch of wrinkles. Those wrinkles are called rugae. There's the word right here. That's plural, R-U-G-A, ruga is singular, and R-U-G-A-E is the plural. It's really just where the mucosal lining is wrinkled up so that when the stomach's empty, it sort of collapses down, and when it fills, the mucosa will stretch out, and those wrinkles will disappear, and that way that the mucosa lining doesn't get distorted or damaged while the stomach's enlarging. We'll see rugae actually in other places in the body where the same sort of enlargement takes place. What's another organ you think would have wrinkles on the inside because it has to expand as it fills? Right, bladder has rugae also. We'll see that actually next week, or next week when we're talking about the urinary system. In the pyloric canal, the rugae tend to be oriented sort of in parallel, acting a little bit as a funnel, so food will go towards the opening down here, which is called the pylorus or the pyloric sphincter. And that's the thing that regulates what leaves the stomach and goes into the beginning of the small intestine, which is called the duodenum, down here at the bottom. In this picture, they show that the stomach muscularis actually has 3 layers. So there's the longitudinal and circular layers that were mentioned earlier for the general structure of a digestive organ, digestive tract wall. And then they have an oblique layer, which is just referring to fibers that are at an angle to the others. So longitudinal fibers are basically running the length of the muscle, length of the organ, and circular fibers are running around it. They will be at essentially 90 degrees to each other. But the oblique fibers are kind of 45 degrees in between the two, giving us just another layer of musculature in there. This figure shows us the basic structure of the wall of the stomach, it's histology. It has, again, the same 4 layers, the mucosa, which has a simple columnar epithelium, its associated connective tissue in the lamina propria, and a thin layer of muscle tissue called the muscularis mucosae. And then it has a submucosa, which is deeper connective tissue. And then it has the muscularis layer, which is, again, for the stomach, 3 different layers. And it has a serous membrane layer on the outside. An important structure in the stomach are the glands that produce the gastric secretions, which are called gastric glands. They are deep invaginations within the mucosa of the stomach lining, and the openings from the surface of the stomach in are called gastric pits. So we can see a bunch of holes or gastric pits all in the surface of the stomach in this drawing. And each one opens up into a gland. They've kind of color-coded the picture, the cells in the picture here. The lighter staining cells of the gastric pit and the gastric surface are just basic mucosal cells. The darker staining ones in the gastric neck and the beginning of the gastric gland are, again, just mucus cells. They secrete the watery mucus. And then the inset in here shows us 3 other types of cells that are at the root of the gastric gland. Now these pictures are drawn representing what you'd see under an electron micrograph, where we can see some differences in the structure of the cell. Under a light microscope, you would just see a bunch of columnar cells. You wouldn't see this sort of detail you see here. At the top of the inset picture, these two things that look like bizarre Pac-Man or whatever are parietal cells. They're responsible for making the stomach acid itself. And then the main type of cell, it's most of what's left over, or what's in here, the kind of tannish cells, these are chief cells. Now, I already mentioned chief cells. Those are the cells that make the enzyme pepsin. They actually make pepsinogen and secrete that into the lumen of the stomach, where it becomes pepsin when it's exposed to the acid. The acid is made by parietal cells, and the enzymes are made by chief cells. So those two cells are making different components, which are each separate until they meet together in the lumen of the stomach. And then when pepsinogen is exposed to the low pH of the stomach acid, it gets activated and becomes pepsin. The other type of cell here at the bottom, called the enteroendocrine cell, or G cell, makes a hormone called gastrin. So they're called G cells to refer to gastrin that's made there. The chief cells are depicted here with vesicles in their apical surface, which represent the secretions that are going into the lumen of the gland and therefore the lumen of the stomach. It's an odd color choice, but the blue things at the base of the enteroendocrine cells represent vesicles also. And the gastrin that they're secreting is being secreted into the lamina propria. Gastrin is a hormone that tells gastric glands what to do. Tells them to make more acid or make more enzymes or make more gastrin. So it's a hormone essentially that's released and has effects on that local tissue, which is technically a paracrine effect. The gastrin can, however, get into blood vessels in the local neighborhood and move to other regions of the stomach and have an effect there, which is a more classic endocrine effect. But gastrin is actually a good example of the three different things that hormones can do, which is autocrine activation. Gastrin released by that G cell will tell that G cell to make more gastrin. They're paracrine because they tell the chief cells and the parietal cells and the other mucous cells to be more active. And they're potentially endocrine because they can move through the bloodstream to other regions of the stomach and have similar effects. But, which is what we see. So those are the ones we see there. Now, unfortunately, the book doesn't have a picture of the parietal cell. So let me pull one up here. And I found a good one Wednesday night. Let me see if I can find it again. A lot of these pictures describe what this is good. A lot of the pictures describe what, yeah, that's fine. These cells do with a little bit more detail than I want to go into. So this image, is a little too small. This was not the one that I found Wednesday night. Let me find a better one. Where did it go? That's big enough. So first off, the picture here is just showing us a very basic sort of blocky cell. And it doesn't have the structure that we saw a second ago in the picture, but this shows us everything that's happening. Now, I don't know why it's labeled stomach lumen down here at the bottom. The stomach lumen, or the lumen of the gland more accurately, is actually out here, where hydrogen ions are getting pumped out of the cell into the stomach. And also chloride ions get out there as well. So stomach acid really is HCl, or hydrochloric acid. Now, hydrochloric acid in solution is just a solution of hydrogen ions and chloride ions. So the fact that the cell doesn't actually make HCl and secrete that as one unit is fine. It just secretes hydrogen ions and chloride ions individually, and when they get into solution, they make hydrochloric acid. Now, the way that happens, it's all kind of interrelated, but let's start with where the hydrogen ions come from. Oh, that's a weird thing about this picture. I don't like this picture. Let me go back. I was trying to find the one that I showed Wednesday night, and I'm not seeing it quickly. This is not it, but it's still a good, actually maybe even a better representation of things. So again, hydrogen ions are pumped out. Now the hydrogen ion comes from, oh, this still does the same kind of thing. Sorry, that's not a good picture either. Internet's kind of bad for these things sometimes. Really wish I could remember where I found it. So a lot of these other pictures that I'm pulling up are not good because they go into, this is the one I wanted. Okay, yay, found it, all right. Again, hydrogen ions and chloride ions each leave the cell individually, giving us hydrochloric acid in the stomach. The hydrogen ions come from carbonic acid. And this picture I was just showing you as I was about to start describing this, I realized that it had this chemistry wrong. And the first one, I don't know how wrong the chemistry was, it just didn't actually write out carbonic acid. But carbonic acid we met in last week, which has to do with how carbon dioxide gets tied up in red blood cells for transport. And here, carbon dioxide combines with water, and thanks to the enzyme carbonic anhydrase, it is made into carbonic acid, which is the opposite of what the name implies. It's putting them together rather than taking them apart. But then carbonic acid dissociates into the hydrogen ion and the bicarbonate ion. The hydrogen ion is pumped out of the cell through a pump that pulls potassium in with the hydrogen that's pumped out. And it requires ATP. It has to hydrolyze ATP for that to happen. So where else have you heard of a pump that pushes a positive ion out, or pushes a positive ion across the membrane in one direction, yeah, which actually is out, and pulls the potassium ion into the cell using ATP as an energy source? Where have you seen that before? Right, so if we just substitute sodium for hydrogen here, we have the sodium-potassium pump that was introduced in AMP1, talking about the membrane potentials. So this is just an altered form of that protein, a different version of the protein that pumps hydrogen out instead of sodium and pulls potassium in. It has to move those two ions against each other. It's the only way that it'll work. But the potassium isn't terribly important for this cell. So it gets pulled in by the pump, but then it just leaks right back out through a potassium channel. And so the potassium just keeps cycling through here because its only purpose is to complete the function of this pump. The potassium itself isn't doing anything for the cell or for the stomach. It just sort of going around and around through the pump and the channel and the pump and the channel and the pump and the channel and the pump and the channel. And every time that happens, ATP gets hydrolyzed to drive that pump. Now, the bicarbonate ion leaves the cell, and it leaves through another transporter molecule that does not require ATP, so we don't call it a pump. But when a bicarbonate ion leaves the cell, chloride ions come in. Now, in the respiratory system, this was happening also. Bicarbonate leaves the red blood cells when carbon dioxide is taken in at the tissue, and then it re-enters when carbon dioxide is leaving in the lungs. But here in the parietal cell, it's just we're making a bunch of it to generate these hydrogen ions for the stomach acid, and so bicarbonate is a byproduct of that, and it leaves the cell. When bicarbonate leaves a cell, any type of cell, whether it's a red blood cell or a parietal cell or anything like that, chloride has to come into the cell. Okay? Just like sodium and potassium or hydrogen and potassium have to be pumped in opposite directions by these pumps in their respective membranes, the bicarbonate transporter only works when chloride moves in the opposite direction. And so chloride gets into the cell and just leaves the cell and gets out into the lumen of the stomach, completing the HCl of hydrochloric acid. The bicarbonate moves out of the cell and into the blood, where it will act as a buffer for the blood to keep blood pH from getting out of control. And that's what it does when it's made by the red blood cell too. But here we see it coming from a different system. This figure gets at the next part of what we want to think about with the stomach, which is how it's regulated. Now, stomach regulation means really making the stomach become more active or less active. So the gastric glands make more acid, more enzymes, and more mucus, and more of the gastrin hormone. The gastric glands do those things, and the muscle layers of the stomach become more active, or everything shuts down. The glands become less active and the muscles relax. And there are three phases of regulation. There's the cephalic phase, meaning what comes from the head, the gastric phase, meaning what the stomach does for itself, and the intestinal phase, meaning what the small intestines do in response to the stomach moving food on into the intestines. The cephalic phase is largely there to prepare you for eating in the future. So if you see or even think of food or smell or taste food, the sensory or cognitive aspects of that are going to activate vagus nerve activity, and so the parasympathetic system will stimulate stomach secretory activity and, to some extent, stomach muscles contracting. Okay. Which is basically to say, if you see, think, hear food, see or think or smell or taste food, I suppose if you hear, fajitas sizzling on a cast iron grill, whatever, those things that clue you into food being present are there to get the stomach started so that you're ready to digest the food. Chances are, if you're seeing or smelling or hearing food, you're going to be eating it soon. Now, marketing departments have figured out that this is how the brain works. And so commercials make you hungry, so to speak, because they stimulate the cephalic phase. When the vagus nerve activates the stomach and you start secreting more acids and your stomach starts contracting more, that activity will seem like you're hungry. Your stomach wants to digest food, so obviously you should put food in it. So if the thought or sight of food from watching commercials is enough, that'll get you to think, oh, I need to go eat something. Or if you're walking across campus and you go that direction over there, what are you going to pass once you get off campus? Burger King, right. And what are you going to notice about the Burger King that's going to activate the cephalic phase? they pump smells out, like the fries or something like that. So you can smell the food, and the smell of the food will activate things, and you go, oh, I should go eat something. It's really just your stomach responding to these basic cues, which were necessary back when we were hunter-gatherers and food was scarce. When we knew we were hunting or gathering food, we knew we were going to eat, so we got hungry. But nowadays, with supermarkets and restaurants and fast food, we can get food whenever we want to. It's just that we're wired to respond to this cephalic phase of things. Now, there is an inhibitory component of the cephalic phase, which is to say certain things can cause loss of appetite, whether it's depression or, just you don't like mushrooms, and so mushrooms just turn you off, whatever. Those sorts of things will decrease parasympathetic activity and therefore inhibit stomach activity. But largely the cephalic phase is about increasing gastric activity in preparation for eating food. Now, the gastric phase has to do with two things, which is the physical presence of food in the stomach, which will stretch the stomach wall out, and the chemical presence of food. So when you're eating food, you're going to have higher concentrations of the macromolecules in your stomach, and those will activate chemoreceptors, as will the rising pH coming from the gastric glands. So the stretch of those stomach receptors will initiate reflexes, really to talk about vagal. That's wrong. That should say vasovagal reflexes, right? No, I guess it's vagovagal. That's talking about axons going up the vagus nerve to the medulla and then back down the vagus nerve to increase things. But technically, if it's going up to the medulla, that's part of the cephalic phase too. Okay. But local reflexes, they're within the submucosal or myenteric plexuses, will directly cause changes to the stomach activity. And then chemoreceptors in the stomach, recognizing the increase in macromolecule concentration or pH, will increase stomach activity too. Now, there is also an inhibitory part to this, which is when the pH gets very low, the book here says below pH of 2. I usually refer to it as below a pH of 1, but either could be correct. Then chemoreceptors will shut off the gastric secretions, because if the pH is getting that low, then you're obviously digesting a lot of food and you need to slow things down, because a lot of that increase in activity is more like a positive feedback cycle. So you need to shut it down a little bit. They have emotional distress down here, which I find it kind of strange to put in the gastric phase because they're calling it sympathetic nervous system activation, which would actually be other aspects of loss of appetite, which will override the parasympathetic system. But I think the reason why they have it down here is because there are potentially some local reflexes or entirely peripheral nervous system components that can lead to this. So I think that's what they're referring to. But it's really hard to separate this emotional distress out from some of the inhibitory aspects of the cephalic phase. And then at the bottom, we have the intestinal phase. The intestinal phase is largely there to keep the stomach in check. Now, there is some excitatory aspects of the intestinal phase. The presence of low pH and digestive food as it gets into the duodenum will say, hey, you didn't quite completely digest everything, and it'll increase stomach activity. But more importantly, in the intestinal phase is going to slow down what the stomach is doing. Now, what the stomach is doing when its muscles are contracting is it's churning the food up inside the stomach. So let me go back to this picture real quick here. These layers of muscle in the stomach, when they contract in a kind of peristaltic way, they push the contents of the stomach towards the pylorus, but only a little bit can come out of the pylorus at a time. And so the majority of the stomach contents sort of churn back the other direction, and you get these back and forth waves of food moving, which helps to liquefy the food. So I tried doing this once in class, and it wasn't terribly successful. But I took a piece of bread and put it in a Ziploc bag that was full of water, not full of water, but half full of water. And I physically sort of mixed it all up. It got disgusting more than anything else, but the idea was the physical churning of the contents of that bag, like the stomach, will help to liquefy the food faster. And that happens. Each time that there's a wave of contraction pushing food towards the pylorus, the valve will open a little bit and the contents of the stomach will come out. But the majority of the contents of the stomach will churn back the other way. Now, you don't want everything in the stomach to just be pushed on into the duodenum at once, because that's going to overfill the small intestine. So the inhibitory activity from the small intestine is really when the intestine stretches out too much, or there's a lot of lipid or acidic stomach contents, which are called chyme. When that stuff moves on, a lot of it gets to build up in the duodenum. Then the duodenum is actually going to, through local reflexes and autonomic connections, slow down the stomach through what's called the enterogastric reflex, which is a negative feedback reflex and slows down what the stomach's doing. The presence of fatty, acidic, or other macromolecule components to what gets into the small intestine will cause the release of intestinal hormones. And they refer to secretin GIP, gastric inhibitory peptide, cholecystokinin, which is abbreviated CCK, and VIP, which is vasoactive intestinal peptide. Those are all hormones that are released from the duodenum that have an effect on the stomach and sort of slow it down. And a lot of that is so that the food coming from the stomach is released slowly into the intestines so that the intestines don't stretch out too much. There's plenty of space in the intestines for all the stomach contents, but the intestines can't stretch out. The space is really because they're so long. And to slow down the release of material from the stomach, these hormones and the local reflexes that cause the enterogastric reflex keep everything under control for the stomach. So really looking at these three phases, the cephalic phase and the gastric phase are largely inhibitory, stimulating stomach activity. And the intestinal phase is mostly inhibitory. Now there's a little bit of excitatory aspects from the intestinal phase, and there's a little bit of inhibitory from the cephalic and gastric phases. But for the most part, cephalic and gastric increase gastric activity, and the intestinal phase decreases it, which is what this figure is getting at. I just want to point this out in the chapter in the book. They refer to the mucosal barrier, which to some degree is referring to the idea of barriers in the innate immune system. It's a barrier that doesn't let things across it. The very acidic contents of the stomach are often used to digest pathogens that might have gotten into our respiratory or digestive tracts. But we need to protect ourselves against our own stomach acid. And so the mucosal barrier has a way of protecting itself, which is that it will neutralize the stomach acids. It releases buffers that try to neutralize the acids right at the edge of the stomach lining. So in the center of the stomach, the acid is very acidic. When we get closer and closer to the tissue of the stomach lining, there are secretions from the mucus cells in the stomach that help to buffer a little bit. Then this little box here talks a little bit about ulcers and when those sorts of buffers become problematic. I described this a little bit ago when I said the churning action back and forth in the stomach. In the book, they address that here under mechanical digestion. And that churning is what's referred to here as a mixing wave. Just sort of a complex peristaltic wave of contraction pushing the food towards the pylorus, and then it mostly churns back the other direction. I already talked about the chemical digestion stuff, which is mostly proteases like pepsin or renin in the infant stomach that play a role there. The next section is about the small and large intestines. I want to concentrate mostly on the small intestine and address how the small intestine increases the ability of that organ to absorb food. And there's really 4 aspects to that. So let me move over here and write these down. Nope, that's not right. Oh, it's got a T in it. All right. There's basically 4 ways that it does this. And the book highlights 3 of them. And I want to talk about all 4. Now, in the book, there we go. After this figure, which has basically 3 parts, in the text, they describe circular folds, villi, and microvilli. And those are 3 of the 4 things that I'm talking about. But those are actually the 2nd, 3rd, and 4th. And I'll explain how they work in a second. The thing that's sort of first in this list is the length of the small intestine. So I quickly opened up a couple of pictures out of this region so I can talk about this. The first one is sort of an overview of the structure of the small intestine. So the small intestine has 3 different regions. I've been talking about the duodenum, which is the beginning, which comes off of the stomach. And it's about 15 centimeters in length or something like that. That's followed by a region called the jejunum, which is highlighted in green here. And then the second half is the ileum. Now the ileum actually ends right here where it connects to the large intestine. So the tube really is getting from point A, where it comes off the stomach, to point B, where it gets to the large intestine right here. And a straight line from point A to point B, let's say that'd be about 20 centimeters long. Okay? But in fact, the small intestine is much longer than that. Now this picture probably is not showing it quite as extensive as it really is. Different sources will tell you that the small intestine is of different lengths, and it doesn't really matter how long it is. It's long. That's the point. Some sources, up until recently, would say that the small intestine is like, I can't remember if it was 7 meters or 7 feet long. It was 7 meters. But a paper that came out recently, and apparently the authors of that paper went on Wikipedia and made some major edits to the page, explained that a lot of the estimates for the length of the small intestine are probably overestimates because of different aspects of how the methodology used to do that. So they suggest that it's shorter than that, but I don't think they actually give a real number for how much shorter it is. But even if it's like half the length, it's still a good length, a good bit more length than a straight line from the end of the stomach to the beginning of large intestine. So just by increasing the length of that tube, there's more tube to absorb things in. Now the other three things are all highlighted in this picture here. In the sort of the drawing part at the top shows the three different parts, and then there are micrographs corresponding to each one. So the circular folds, which in Latin are called plici circularis, if you see that, are kind of just the inside surface of the mucosa scrunched up inside. So with the micrograph, you can see the mucosa that's sort of wrapped over and over these folds. If you could stretch it out flat, it would become much longer. Okay? Now, I'm a child of the 80s, so scrunchies were a big thing when I was a kid. I didn't wear them, but I knew a lot of women that did. And so that's what I always picture here, a scrunchie, just an elastic, a fabric-covered elastic that's scrunched up. And if you were to pull the elastic out as far as it would stretch, the fabric covering of it would flatten out a little bit. That gives you an idea of what the circular folds are like. Now, on top of the circular folds that we can see in the micrograph, the mucosa isn't just flat. There's actually little folds inside of that. And those folds are of the mucosal lining, the epithelium, where it wraps around these finger-like extensions out of the mucosa called villi. Villus is singular, villi is plural. And so instead of just having a flat sheet of cells, if we went from this side of the picture straight across, we might have 50 cells going from one side to the other in 3 dimensions. But instead, wrapping the mucosal epithelium around this finger-like extension gives us, I don't know, maybe a factor of 5 or 10 more cells. So instead of just 50 going along the base of the villus, wrapping around the entire villus, there might be, what would that be, 500 or so cells more, okay? Just increasing that. Now within the villus, there are blood vessels. So we have a intestinal arteriole coming in and an intestinal venule coming out with capillaries in between, and then a lymphatic capillary called a lacteal that's sort of in between all of that. The lacteal is there to absorb is there to absorb lipids from our diet, and then the amino acids and carbohydrates that are absorbed go directly into the blood supply. And then the lipids travel through the lymphatic system and eventually get introduced into cardiovascular circulation associated with lipoproteins, which help them dissolve in the blood. Okay. And the transporting of dietary lipids is one of the functions of the lymphatic system that came up back then. But the structures that are absorbing things into those two circulatory streams are inside the bulk of the villus. And here we see a higher power micrograph showing us what's happening in the mucosa with the cells wrapped around this finger-like extension. That one finger-like extension that I'm pointing out here is going to be one of these little fuzzy parts on the surface of the circular fold that we see here in this lower power micrograph. The fourth aspect of how this works is the apical surface of the cell. So each of these cells here, on their apical surface, the cell membrane is not flat, but instead has a bunch of microvilli sticking up out of it. And those microvilli are what we call the brush border. They're sort of a fuzzy edge to the apical surface of the cells. And the brush border enzymes are positioned right around there to help break down the nutrient molecules to their final form before they get absorbed. The micrograph at the bottom here is an electron micrograph showing us the apical membrane of one of these cells with microvilli highlighted in the picture. So again, there are four aspects of how the small intestine increases the absorption of nutrients by increasing surface area. If you have a longer tube, then there's more surface area inside to absorb things. And then if that inside surface is scrunched up in these circular folds, you just have more mucosal lining there for it. And if the mucosal epithelium is wrapped around these villi, that's another layer of increasing surface area. And then at the actual apical surface of those cells, instead of just being flat, you have much more cell membrane to absorb things across because they're folded over and over in these microvilli. And that just optimizes the small intestine's ability to do its job. The book then has a list of components that play a role in intestinal glands secreting important parts. I'm not going to go into any detail there. It mentions the mucosa-associated lymphatic tissue that came up in the lymphatic system, intestinal mold. Okay. This picture looks very similar to the what sort of link for? Peristalsis picture that I showed you earlier, but this is showing a different type of mechanical digestion / propulsion called segmentation. So there are circular fibers that pinch off at regular intervals, separating the contents of the small intestine into these little packets of food. And they've color coordinated it so that we see yellow, teal, whatever that is, back and forth. Then in the middle of each of those little segments, the circular fibers constrict, and the ones that are constricted in this first picture relax, and we get the segments mixing. So half of this teal one and half of this yellow one come together, making a mixture of the two. Okay. And then the same thing happens again, and circular fibers in the middle of the segment contract, and the ones that are already contracted relax, and we get remixing of that segment. Now at this point, they're coloring it just green because we're mixing these two colors so much that they're all the same color, so to speak. But these segmentation waves just keep going back and forth and mixing things up. Added to the segmentation is a peristaltic movement. So while it's sort of going back and forth between pinching off in the middle of alternating segments and that sort of thing, the longitudinal fibers are also kind of helping everything move down the length of the small intestine. And that's a peristaltic wave moving these segments, which are also getting mixed up to help the food break up a little bit more. Then it goes into chemical digestion of the small intestine. There's a lot going on there. I've mentioned the main things that I want to, which are pancreatic enzymes and brush border enzymes. I went into some detail about lactose intolerance, and they have a little box of that here. Then the chapter goes on to talk about the large intestine, and this, you can get a lot of this anatomy straight from the book. It starts with the cecum, and then the colon, which is really the ascending, transverse, descending, and sigmoid portions, and then it ends in the rectum, and then opens up at the anus. That's all of the anatomy there. The histology picture is kind of similar to the one in the stomach, except that it doesn't have very complex glands. All that the intestinal glands are doing in the large intestine is making a water-rich mucus, which helps to lubricate the surface of the large intestine so things can move on there. There's not a comparable picture of the histology of the small intestine in the book, but it's not going to be that, well, I should say, it's probably going to be about halfway between the gastric glands in the stomach and simple intestinal glands like this. They're slightly more complex, but not quite as complex as the stomach. And then this little inset here is showing us a goblet cell, and there are goblet cells throughout the intestinal lining. Not just the large intestine, large and small intestine. Here's showing some other finer structures of the anatomy there. Talks about the bacterial population that lives in your large intestine and some other aspects of digestion. And it talks about the defecation reflex, how when the solid matter, the feces, get to the end of the colon, they move into the rectum, and as the rectum stretches, there's a reflex through the parasympathetic system that will cause muscle contractions. It'll push the feces out of the rectum. And then there are two sphincters at the opening of the anus called the internal anal sphincter and the external anal sphincter. The internal sphincter is smooth muscle. The external sphincter is skeletal muscle, which means you don't have voluntary control over the internal sphincter. It's part of the reflex. When your rectum starts pushing the feces out, the internal sphincter will relax so the feces can move. The external sphincter, however, you do have voluntary control over. If you don't want to defecate, then you won't. Up to a point. There's a point at which you probably don't have much control over anything because you can't just completely not void your bowels. The next section is about accessory organs, and here it's really talking about the liver, pancreas, and gallbladder. Personally, I would have maybe said pancreas, liver, and gallbladder, or liver, gallbladder, and pancreas, but the book here actually put them in that order. The liver is really just a big blood filter. It does a number of different things. For the digestive system, it filters digestive things out of the blood, which we'll get to in a second, and forms the digestive juice called bile. Now, what is in bile are several components, including what are called bile salts, which are really the emulsifying agents. They'll get in and break lipid molecules apart from each other, sort of defeat the hydrophobic interactions, so that enzymes can get in there and break them down. You've already learned about one bile salt. It's called bilirubin. Do you remember that being introduced? Where's bilirubin come from? When did we talk about that? Anybody remember? I think I heard somebody say it.
In the blood.
In the blood, right. What component of the blood becomes bilirubin? Do you remember? It's the heme molecule. When red blood cells are broken down, pretty much everything gets recycled except for the heme molecule, which gets repurposed. It gets to the liver, and the liver modifies it from heme into bilirubin, and the bili part of that word is referring to bile. And it's secreted with the other components of bile into the small intestine, helps to emulsify lipids in our food so that we can break them down. And then we absorb that back into our bloodstream. And so as the liver's filtering blood that's coming from the small intestine, it'll recover those bile salts, like bilirubin, and make more bile. So when you have lipids in your system and you're emulsifying them with bile, you'll be reabsorbing that bile, it'll get back into the system, and it'll cause the production of more bile. Not all of the bile salts get recovered. Not everything gets reabsorbed. So anything that stays in the small intestine moves on into large intestine and becomes a component of feces. Bilirubin, as it passes through the intestines, gets modified and changes into a different molecule. Actually, it makes a few changes. At the end, it becomes a molecule called stercobilin. Again, it has bil as part of its root, referring to its presence in bile. Stercobilin is the pigment molecule that gives feces its particular color. So it's a sort of tan to brownish colored pigment that's found in feces. The color of feces can indicate liver trouble. So if there's not enough bile being made and there's not enough stercobilin in the feces, if it's the wrong color, it points to a problem with the liver. I'm going to skip over the pancreas. I'll come back to it in a second just to say a few things, but I want to get to the gallbladder because that's really related to the liver. So we see in this picture the liver taking up most of the background space where bile is made. That bile accumulates into ducts called the left and right hepatic ducts. They drain through the common hepatic duct into the common bile duct, which then connects to the small intestine and releases bile into the small intestine. If you are not presently digesting food, you don't need bile in your small intestine. So there's a little sphincter at the end of the bile duct that closes it off. And so when the liver's making bile, if there's no need for bile, it backs up in the common bile duct and backfills through the cystic duct to fill in the gallbladder. Cyst, as in cystic duct, is a word that basically means bladder. It has a few other meanings, but the cystic duct is the gallbladder duct. The gallbladder will store the bile until it's needed at the beginning of the next meal. The main cause of contraction of the gallbladder is a hormone, which I mentioned briefly earlier, but let me just write it down, the name here, called cholecystokinin, which is abbreviated CCK, cholecystokinin. That word literally translates to mean gallbladder movement. Okay, I just told you cyst, as in the cystic duct, refers to a bladder. Chole is a Greek word that means bile. And then kine is the Greek root that means movement. It's the root of the word cinema, with the K being changed to C. So cholecystokinin means bile bladder movement. And it's an enzyme, a hormone that causes that. Cholecystokinin will also increase pancreatic enzyme secretion. Cholecystokinin is going to be released from the duodenum when hypertonic chyme, or lots of macromolecules, move from the stomach into the small intestine. And so it wants all of the bile that's been stored up in the gallbladder to be released so that you can emulsify the lipids. And then it wants enzymes from the pancreas to break down those lipids and carbohydrates and proteins in that food. Another important enzyme from the duodenum that was mentioned briefly when I was showing you that stuff with the intestinal phase, secretin will be released in response to the pH of what's being released from the stomach. And it increases liver production of bile and increases pancreatic bicarbonate secretion. Now, actually, bile has some buffering capabilities too, but the bicarbonate can be released from the pancreas, and bicarbonate or other buffers in bile are there to neutralize stomach acid as it moves into the duodenum. Okay. So let me add something to this. So chyme is the name of the watery substance that's in the stomach, and it moves into the small intestine. When it's hypertonic, that means it has a high concentration of nutrients in it. molecules dissolved in it. So when there's a lot of molecules moving on from the stomach and the small intestine, it causes cholecystokinin to be released, which has those effects on the gallbladder and the pancreas. When stomach acid gets into the small intestine, that causes secretin to be released, sorry, I spelled that, to be released, and the liver produces more bile, and the pancreas secretes bicarbonate, which will help to neutralize the stomach acid as it gets into the small intestine. Now, I'll come back to this slide if you didn't quite write it all down, but I just want to, oops, not that picture. Go back here and point out the pancreas. I'm not going to really say too much about the pancreas because we largely talked about its structure in the endocrine system because it's responsible for releasing insulin from the pancreatic islets. So when we were looking at the structure of the islets, I also talked about the darker staining tissue around that, which is the exocrine tissue, which is what's making the enzymes and the bicarbonate being secreted in the small intestine. Okay. So the book in this chapter rehashes that a little bit, but I covered it in the endocrine system stuff. So that's what I wanted to say about the accessory stuff. I want to actually move, well, the next part of the book, next part of the chapter is basically the enzyme stuff that I talked about earlier. I want to take a second to move into the next chapter. We will not have a chapter assignment set for this metabolism and nutrition chapter, but I just want to mention a couple of things that have to do with the digestive system. Now, in this chapter, they go over a lot of different things about metabolism. Metabolism is really just all of the chemical reactions that take place in your cells. I just want to concentrate on carbohydrate metabolism, and in fact, I want to concentrate just on cellular respiration. So I introduced cellular respiration, remember that from last week, C6H12O6 plus 6 O2s yields 6 CO2s plus 6 H2Os. That's the cellular respiration reaction. What it is it's a chemical reaction to lead to energy extraction from nutrients so that we have energy to make ATP. This figure is a nice overview of that process. So it starts with glucose being broken down in glycolysis, which happens in the cytoplasm of cells. Glucose is a 6-carbon sugar, and it's broken down into 2 3-carbon sugars called pyruvate or pyruvic acid. The pyruvate moves into the mitochondria, into the matrix, which is the center of the mitochondria, where it gets modified a little bit. One of its carbons comes off as carbon dioxide, and the other 2 carbons are attached to something called coenzyme A, or CoA, where it then gets introduced into the Krebs cycle, or citric acid cycle, or it has a number of different names. This is a series of reactions that completely break down the carbon molecules added to them. And I'll talk about that in a little bit more detail. That will eventually break the carbon molecules all the way down to carbon dioxide. Okay. So we have C6H12O6 at the beginning. We have 2 CO2s at this point and 4 CO2s at this point, so that for a total of 6 CO2s that come out of that. In doing that, we produce a little bit of ATP, some in glycolysis and some in the Krebs cycle, but most of the energy gets harvested as electrons that then, in the mitochondrion, are used at the inner mitochondrial membrane through a series of reactions called the electron transport chain that will lead to the production of a lot of ATP. At the end of the electron transport chain, the oxygen that's necessary for this to happen gets reduced so that it can combine with hydrogen ions and give us H2O. Okay, so one C6H12O6 gives rise to 1, 2, 3, 4, 5, 6 carbon dioxides, 2 coming out in the transformation of pyruvate, and 4 coming out in the Krebs cycle for each glucose molecule. And then we generate 6 water molecules when 6 oxygen molecules are present because of the electrons that are present in this process. Now, the electrons that are harvested here, we have to maintain a balance. And so for every electron, we need a proton. And what's another name for a proton? It's on this picture right here. What do we call one proton with 1 electron together? Nope. It's a particular type of atom. What type of atom has 1 proton? Hydrogen. So if we strip an electron off of a hydrogen atom, we end up with a hydrogen ion, which is just that proton. Okay. So these H pluses down here refer to those hydrogen ions, which are basically the protons that offset the electrons here. So we harvest all these electrons out and drive this electron transport chain, and hydrogen ions, or protons, get combined with the reduced oxygen to make water. Okay. In this part of the chapter, whoops, too far, they explain these things in a little bit more detail. You don't need to understand the extreme detail of what these pictures are going into. For the glycolysis, all that I really ask you to understand is a 6-carbon molecule gets broken down to 2 3-carbon molecules at the end. 2 ATPs are necessary to get the process started, and we generate 4 ATPs from that. So a net gain of 2 ATPs for each molecule of glucose. We also get some electrons out of that here in the middle portion. Then this is a kind of complex figure, but it shows glycolysis up there at the top. And when oxygen is present, we go to the Krebs cycle and the electron transport chain, which is basically out of the picture I showed you earlier. When oxygen is not present, then the pyruvate ends up getting changed into lactate, or lactic acid, which is a waste product. So that's about all you need to get out of that. Oxygen present, we finished the story. Oxygen is not present, then the pyruvate gives rise to lactic acid. This is the Krebs cycle. I do not want you to learn this in a lot of detail, but it does show you that there's just recurring reactions here. A 4-carbon atom and a 2-carbon atom come together to give us a 6-carbon atom, which is modified to a different 6-carbon atom, which is modified to a 5-carbon atom where it gives off 1 carbon and carbon dioxide, and then it gives off another carbon and carbon dioxide, and it's a 4-carbon atom, which is modified to a different 4-carbon atom, to a different 4-carbon atom, to a different 4-carbon atom, to a different 4-carbon atom that combines with a 2-carbon atom, I keep saying atom, molecule, 4-carbon atom, 4-carbon molecule, 2-carbon molecule, et cetera, et cetera. And it just repeats over and over again, the same reactions. We get rid of carbon as carbon dioxide at 2 points. We get some energy in the form of GTP, which is an analog to ATP, as well as pulling off some electrons, which we see in a few different places here. Don't worry about the specific chemistry here. I just want you to see that it's just a series of repeating actions, repeating reactions. This figure shows what's happening with the electron transport chain in the inner mitochondrial membrane. Let me enlarge this. Okay. So these kind of maroon-looking things are membrane proteins that pick up the hydrogen ions and pump them across this membrane using the energy from the electrons. Eventually, those electrons will reduce oxygen so that we can make H2O. All of the hydrogen ions that have been pumped across this membrane want to get back across to the other side, and the only way for them to do that is to go through this big enzyme here, which is the ATP synthase. So it uses the movement of hydrogen ions across the membrane to physically cause ADP to be phosphorylated into ATP. And that's where the hydrogen ions come from that get to be part of the water molecule. So this is just a big pumping station that drives ATP synthesis. And then this is sort of the same as the first picture, but now they're putting specific numbers in. For one glucose molecule, you get 2 ATP out of glycolysis, 2 ATP out of the Krebs cycle, and up to 36 ATP, 34 ATP. Something looks wrong there, but the grand total, generally speaking, is thought of as 36 to 38 ATP out of all of this happening. So we can make a lot of ATP when oxygen is present from the energy stored in a molecule like glucose. Now this section goes on to talk about other carbohydrate metabolism processes, but all I'm interested in is the production of ATP from glucose, the cellular respiration that I introduced last time. And some of these figures get into a bit extra detail that I don't want you to get bogged down to, but it does sort of tell you the story of how it all works there. There's also a section here on nutrition, which by itself doesn't have a whole lot to say, other than just mentioning some of the micronutrients that we need in our system. But what I want to introduce is a special kind of macronutrient, which means macromolecule like lipids, carbohydrates, and proteins, which are called essential nutrients. Now, there are essential amino acids. There are 20 amino acids that we need in our system to make proteins. And the ones that are listed here that are essential amino acids, we have to get from our diet. If we don't get these in our diet, we can't make the proteins we need to in our body. The non-essential ones, either we can make from other things or we can make when essential amino acids are present, which I think is what conditional means. If you don't get these amino acids in your body, then you can't make the proteins you need. All animals have the same basic amino acid requirements. So if you want to get a definite source of amino acids and not have to worry about it, where it's coming from or which one you're getting, eat other animals. That's the point of carnivorous diets. So you are an animal, you eat other animals, you get the proper protein load taken care of. If you're not too crazy about that approach, if you don't want to eat animals, then you just have to be aware of your diet and get the correct essential amino acids. If you are a vegetarian or a vegan, you're probably aware of this, or you're really, really sick, that you should eat things like rice and beans, or any grain and legume combination. So quinoa and lentils, or I guess pasta with beans, which is the same thing as rice and beans. But anyway, those sorts of foods in the proper combinations will give you the essential amino acids you need. If you are, say, a completely fruit and vegetable type of vegan, then you're not going to get what you need to out of fruits. You have to eat the proper combination of vegetables out of that. Okay? But it's not impossible to do if you have reasons for being vegan or vegetarian. Now, the other type of essential nutrient that I want to mention are essential fatty acids. Now, when I do a image search for essential fatty acids, oh, actually, well, you get a lot of pictures like this that show you foods that are good sources of these types of fatty acids. What I want to show you is the structure of fatty acids. Actually, let me go here. So fatty acids are carbon chains with a special thing on the end. So the gray dots here are carbons, the white dots are hydrogens, and then these red things at the end are oxygens. So at one end of a fatty acid, we have a carbon atom double bonded to an oxygen and single bonded to an oxygen that's also bonded to a hydrogen. So that's COOH. You'll often see that. That's the alpha end of the fatty acid. The opposite end is called the omega end. Fatty acids can be saturated, which means that there are only single bonds between all of the carbons. Or they can be unsaturated, which means that there's at least one double bond in that chain. This double bond here is in a position that's not terribly important to us. The essential fatty acids are ones that have double bonds in very specific places. Does anybody know what the name of the common essential fatty acids are? And the fact that I mentioned the omega end is part of how we name them. Anybody know what they are? You ever heard of omega-3 or omega-6 fatty acids? Those are essential. Now, if we start from the omega end, this is the first bond, this is the second bond, this is the third bond, and that's where we see a double bond. So this is an omega-3 fatty acid with a double bond in the third carbon-carbon bond. Or the one above it, 1, 2, 3, 4, 5, 6 bonds in from the omega end. This is an omega-6 fatty acid. The omega-3 fatty acid that's here is alpha-linolenic acid, and the omega-6 is just linoleic acid. Those are two essential fatty acids that we need to get in our diets. Does anybody know where we get omega fatty acids from? Omega-3 and 6? What?
Vitamins, fish.
Not vitamins.
Well,
necessarily.
I don't know.
So we need to get them from sources of oils, and fish oil is a great source for that. But again, if eating animals is not your thing, you'd have to go to somewhere else. Anybody know of non-animal sources of? Yeah, flax seeds oil is a great source of that. My mom's one of those people that is really too old for the internet, and so she learns of things on the internet, and she says, "Oh, this is what you're supposed to do." And I don't know if she still does it. She might, but for a long time, whenever I visited her, she came to visit me. She had a little bag of flax seeds, and every morning with her cereal for breakfast, she'd dump a bunch of flax seeds on that and eat that as an additive to her morning breakfast. And that's one way to do it. Or you can get flaxseed oil and cook with that. Actually, I don't know if cooking with flaxseed oil is a good idea. Heat might actually mess up the fatty acid structure.
What happens if you don't eat fish? Like I don't eat fish, and I don't take those vitamins. So.
Well, you probably eat vegetable sources that have some of those in them.
Yeah, okay.
But primarily the reason why these essential fatty acids are important is because they're necessary for making membrane lipids. So if you don't get them in your diet in sufficient numbers, then cell membranes will not function 100%.
How do you know? Will the doctor tell you?
I'm actually not sure, and I don't know that.
Do you feel tired? Or?
I would bet that there's. I mean, if it's integral to just you have never gotten that in your diet, then it's probably kind of foundational in your behavior or whatever. But if you normally got it and then suddenly you stopped taking it in, then it would be obvious. No, it's bad either way. It's just I don't know if anybody would notice it if you've never gotten it in your diet at all. But I think sunflower oil is another good source. So, and that's actually probably used. Yeah, that's used a bit in sort of normal cooking and food production things. So I wouldn't worry too much, but maybe you should get some flax seeds and start putting them on your breakfast in the morning. Well, if you don't like fish, then flax seeds probably will taste better. But if you don't like,
Yeah.
if you're not eating fish because you think fish is gross or something like that, then go with the flax seed thing. If it's not a taste issue with fish, then get fish oil supplements, which I think taste a little fishy. So.
Is that bad? It's like the aftertaste of them is, I think, not the
I've never
initial
taken
taste.
them, so I don't know. I just eat the
My mom
fish.
takes them, and, like,
It's a lot
it's
easier though.
Like what
Yeah.
kind of fish? Just any fish? Like,
Oily fishes I think are better, like salmon or.
I'm sorry.
I don't know if tuna is necessarily the
Oh, I
best
eat
source.
tuna.
I don't know if tuna
Oh.
is. But maybe
Okay.
look on the container. If you do eat tuna,
Yeah, that's
if
true.
it says something, they'll market it that way if it is a component of it. Any other questions? So I wanted to just finish off the digestive system section by mentioning these essential nutrients, the essential amino acids and essential fatty acids, just to introduce those as things that you should get in there, into your diet. I'll probably include that information in the presentations when I do that. Whenever I get it up there, it'll be up there eventually. But I have recorded today's stuff, so most of what I've gone over you can catch again from the YouTube channel.